MCQ and full zone for self-assessment. Then MCQs are there that are from beginner level to expert level. And also student can book a personalized counseling session in which they will have the direct interaction with the counselor. Then also we provide some subscription plans starting from 3-month plan to 12-month plan. So basic plan is of 3 months, standard plan is of 6 months, and premium plan is of 12 months, which includes all the services that is provided by Auricle, like pre-recorded class, live doubt clarification, one-to-one personalized guidance, notes, quick revision, examination module. And this premium plan cost around 50,000, and also for instant 20% discount, you can use my code, that is Aanchal 20. So, internal exam marathon series has some features like rapid revision to provide volatile facts. Will be discussed. We also covered MCQ, and also we assure top note grades in the examination. So, in today's session, we're going to discuss some very important topic of the respiratory system like hypoxia, neural regulation of breathing, surfactant, difference between restricted and obstructive disease, and dead space, and lung volume and capacities. So let's proceed with the first topic, that is surfactant. So what is surfactant? So surfactant is a surface acting material or agent that is responsible for lowering the surface tension of fluid. Okay. So please remember that surface is a chemical substance that is main aim to lower the surface tension. Okay. So surfactant that lines the epithelium of alveoli. Okay. So basically this surfactant is there to line the epithelium of the alveoli, okay, in the lungs as a pulmonary surfactant, and here it decreases the surface tension of the alveolar membrane. Okay. Now, talking about the source, that from where the surfactant come. So, talking about the source of secretion, there are two main sources. First, type 2 alveolar epithelial cells, and second we have Clara cells. Okay, so type 2 alveolar epithelial cells are found in lungs, which are known as surfactant secreting alveolar cells, and the another name of these cells are pneumocytes. Okay, so student, please remember the name of these cells, because usually the question in the MCQ asks from this. Okay, so these pneumocytes are having a very important characteristic feature that there is a presence of microvilli on their alveolar surface, okay, which aim to decrease the surface tension. Okay. Now, Clara cells are situated in bronchioles. The Clara cells are situated in bronchioles, and these cells are also known as bronchiolar exocrine cells. Okay. So, the another name for Clara cells is bronchiolar exocrine cells. Okay. Now, talking about the chemistry means of what they are made up of. Okay, so surfactant is a lipoprotein complex. It is a lipoprotein complex which is formed by especially phospholipids, protein, and some ions. Okay, so talking about phospholipids, so phospholipids form majorly, that is around 75% of the surfactant, and the very important phospholipid is DPPC. Okay, then other lipids are also there, like other lipid substances of the surfactant are triglycerides, then phosphatidylglycerol, and proteins are also there. So talking about the proteins, so protein are surfactant protein A, surfactant protein B, C, D. Okay, so these are all the components from which the surfactant is synthesized, right? Okay, now next thing, that which factors are very, very necessary for the formation of this surfactant. Okay, so for the formation of surfactant requires many substances. Okay, as we have discussed, protein, ions, phospholipids are there, but formation of tubular myelin required a very important thing, that is DPPC and PG. PG is phosphatidylglycerol. Okay, and also hydrophobic proteins are there that we have discussed, that is SP, B, and SPC, and also the formation of surfactant film requires SPB and SPC and PG. Okay, and also one more substance that is also present that also play a very important role in the formation of surfactant, that is glucocorticoid. Okay, now talking about the main thing, that what are the main function of surfactant. So one by one we're going to discuss that. First is, as we have discussed, the first function of the surfactant, that it reduce surface tension. In the alveoli of lungs, right? And by doing this, it prevent collapsing tendency. Okay. Now, what is another function? The another function of surfactant, that this surfactant is also responsible for stabilization of alveoli, which is necessary to withstand the collapsing tendency. Okay. Then another thing, that it also play in the inflation of lungs after birth. So basically what happened, the secretion of surfactant begins after third month in the fetal life, and until birth, the lungs are solid and not expanded. Soon after birth, the first breath start because of the stimulation of respiratory center, that is caused by hypoxia and hypercapnia. And also, as we know, the respiratory movements are attempted by the infant. The lungs tend to collapse repeatedly, and the presence of surfactant in the alveoli prevents the lungs from collapsing. Okay. Then finally, the another function of surfactant, that it provide role in defense within the lungs against infection and inflammation. Okay, so these are the main function that is performed by surfactant. Okay, now next we have the another important topic of respiratory system, that is lung volume and capacity. So what is lung volume and capacity? So as we know, pulmonary function test or lung function test are very useful in assessing the functional status of the respiratory system, right? In both physiological and pathological conditions. So lung function tests are based on the measurement of volume of air that is breathed and breathed out during quiet breathing and forced breathing. Okay. So for that, we carried out the test that is known as PFT or spirometry. Okay. So in that, what we do, we actually measure the lung volume and capacity. Okay. So one by one, we're going to discuss. So here we're going to discuss about the static lung volume first. So in static lung volume, we have tidal volume. Then we have inspiratory reserve volume. Then we have expiratory reserve volume. And finally, we have residue. Okay, so one by one, we're going to discuss about all these lung volume. So first, we have the tidal volume. So what is tidal volume? Tidal volume is the amount of air that is breathed in and breathed out in a single normal quiet respiration. Okay, and total volume that signifies the normal depth of breathing is 500 ml. Okay. Now next we have inspiratory reserve volume. So what is inspiratory reserve volume? It is the additional volume of air that can be inspired forcefully after the end of normal inspiration. Okay. Suppose I perform normal inspiration like what we do in quiet breathing. Okay. Then whatever the force amount of air that I'm inhaling after forcing, like this, that is inspiratory reserve volume. Okay. So it is around 3300 ml. Okay. Then next we have expiratory reserve volume. So what is expiratory reserve volume? It is the additional volume of air that can be expelled out forcefully after normal expiration. Suppose I am doing normal expiration during quiet breathing, but if I do like, so this will be ERV, that is expiratory reserve volume, and it is 1000 ml. Okay. Now, what is residual? Residual means the thing which has left. Okay. So, residual volume is the volume of air that remaining in the lungs even after forced expiration. Okay. So, normally lungs cannot be emptied completely, as we know, because if the lungs will get emptied completely, so they will collapse. Okay. So normally lungs cannot be emptied completely even after the forceful expiration. So some quantity of air always remain in the lungs after forceful expiration, and normal residual volume is about 1200 ml. Okay. Then next we have lung capacities. Okay. In this we have inspiratory capacity. Then we have vital capacity. Then we have functional residual capacity and total lung capacity. Okay, so one by one, we're going to discuss about all of them. So what is inspiratory capacity? It is the maximum volume of air that is inspired after normal expiration. Means at the end of expiratory position. Okay, so for the formula of its calculation, we can also determine the inspiratory capacity as tidal volume plus IRV, inspiratory reserve volume. So tidal volume is 500, inspiratory is 3300. So it will be around 3800 ml. Okay. The next we have vital capacity. So what is vital capacity? It is the maximum volume of air that can be expelled out forcefully even after taking a deep inspiration. Okay. And also for determining vital capacity, we have a formula that is inspiratory reserve volume plus tidal volume plus expiratory reserve volume. So we can determine that IRV is 3300, our tidal volume 500, and this is 1000. So it will be around 4800 ml. Okay. Then we have FRC, that is functional residual capacity. So functional residual capacity is the volume of air remaining in the lungs after normal expiration. Okay. And also for the determination of FRC, again we have a formula that is ERB plus RB, and it is 2200. Okay. Then finally we have TLC. For TLC is the volume of air present in the lungs after a deep maximum inspiration. And for TLC, we have formula IRB plus TB plus ERB plus RB. And it will be around 6000 ml or 6 liters. Okay, so this is all about the lung volume and capacities. Now, next we have the another important topic, that is difference between um, restrictive and obstructive disease. Okay, so before proceeding, we should know that what is obstructive and what is restrictive. Okay, so obstructive means the disease is the abnormal respiratory condition that is characterized by difficulty in expiration. Okay, so this is difficulty in expiration. And what is restrictive disease? This is also an abnormal respiratory condition which is characterized by difficulty in inspiration. Okay, so we can see the difference also, that here the obstruction means some kind of resistance. Okay, so here there will be reduction due to resistance, there will be reduction in airflow, and shortness of breath will be there, and the air will remain inside the lungs after even the forceful expiration. Okay, and restrictive means some kind of restriction is present, right? So this is also characterized by reduction in the lung volume, so difficulty in taking air inside the lungs due to stiffness inside the lung tissue or chest wall cavity. Okay, so various examples are there, like obstructive lung disease are like COPD, asthma, bronchitis, or restrictive lung disease. We have interstitial lung disease, scoliosis or neuromuscular cause, or marked obesity. Okay, so this is all about the difference between obstructive and restrictive disease. Now, next we have another important topic, that is dead space. Okay, and please remember all the students, and usually the question from dead space comes in the examination. Okay, so what is dead space? So dead space defined as the part of respiratory tract where gaseous exchange does not take place. Okay, so I'm going to write that it is the part of respiratory tract where gaseous exchange does not take place. Okay, and air present in the dead space is known as dead space air. Okay, so here we have two types, that is anatomically dead space and another we have physiological dead space. So what happens in anatomical dead space? Basically, this kind of dead space extend from nose to terminal bronchioles. Okay, so till here it will remain, starting from nose to terminal bronchioles, and it includes nose, pharynx, trachea, bronchioles, branches of bronchi up to terminal bronchioles. Okay, and these structures serve only as a passage of air, move for the air movement, and gaseous exchange does not take place in these structures. Okay, whereas talking about the physiological dead space, so physiological dead space includes dead anatomical space plus two additional volume. Okay, so what happens here? Air in the alveoli. Okay, so air in the alveoli which are non-functional in some respiratory disease. Alveoli do not function because of dysfunction or destruction of alveolar membrane. Okay, so also air in alveoli which do not receive adequate blood flow. So there also gaseous exchange will not take place during inadequate blood supply. Okay, so this is all about the anatomical and physiological dead space. Okay, now if we talk about the normal volume of dead space, so normal volume of dead space is around 150 ml, and under normal condition, physiological dead space is equal to anatomical dead space. It is because all the alveoli functioning and all the alveoli receive adequate blood flow in normal condition, and physiological dead space increases during respiratory disease which affect the pulmonary blood flow of the alveoli. Okay, so this is all about the dead space. Now, next we have the neural regulation of respiration, and also this is the very important question for the examination. Okay, so talking about the regulation part. Okay, so as we know, respiration is a reflex process, right? But it cannot be controlled voluntarily for a long period of time. Okay, so like if I or try also, then still I can hold my breath only up to 40 seconds, not more than that. Okay, so regulatory mechanism for quiet breathing is usually controlled by nervous or chemical mechanism, but here only we're going to learn about the neural regulation of respiration. Okay, so here we're going to talk about the some centers like medullary center and pontine center. Okay, but first of all, we should see that the respiratory cycle. So DRG means dorsal respiratory center. Okay, or dorsal respiratory group of neuron that is that produce inspiratory ramp signal, from where the sensory neuron mainly to diaphragm is the muscle of inspiration. So there will be contraction of inspiratory muscle, inspiration occurs, then switch off ramp signal will occur. That switch off center is present in pontine, and there will be elastic recoil of lungs and chest wall, and expiration finally take place. Okay, now one more thing that you all should remember, that inspiration is always an active process and expiration is always a passive process, means it does not require energy. But in case of forceful expiration, if there is forceful expiration is occurring, so that time it will also become this active. Okay, now we're going to talk about the centers. So here we have medullary center and pontine center. In medullary, we have dorsal group of neurons, and here we have pontine group of neuron, ventral group of neuron, and for pontine center, we have apinistic center and pneumotaxic center. Okay, so one by one, we're going to talk about them. So what is DRG? DRG is dorsal respiratory group of neuron. Okay, so dorsal respiratory group of neuron are diffusely situated in the nucleus of tactus solitarius. Nucleus of tactus solitarius. Okay, which is present in the upper part of medulla oblongata. And okay, and these neurons are collectively known as inspiratory neurons. Okay, so in DRG we have inspiratory neurons only, so that the process of inspiration can be occurred. Okay, whereas we talk about the VRG. So VRG is the ventral respiratory group of neuron, which is situated in nucleus of ambiguous and nucleus retroambiguous. Okay, and here mostly the expiratory neurons present. Okay, so ventral respiratory group of neuron are present in nucleus ambiguous and retroambiguous, and these two nuclei are situated in the medulla oblongata, anterior and lateral to the nucleus of tactus solitarius. And usually the expiratory neurons are present in VRG. And also, if we look, like earlier it was said that expiratory neurons are present, but according to the new experiments and invention, it is said that inspiratory neurons are also present. Okay, now what is the function of VRG? So normally ventral group neurons are inactive during quiet breathing and become active during forced breathing. Okay, because as we know that expiration is always a passive process, but if forceful breathing is there, then only at that time it will become active process. So in for the process of expiration, the VRG stimulation whenever required, but if the expiration is an active process, then at that time only the VRG get activated. Okay, now for pontine center, we have two different center, that is apinistic center and pneumotaxic. So this center, apinistic center, is present in nuclei reticular formation of lower pons, and the pneumotaxic center is situated in the dorsolateral part of reticular formation in upper pons. Okay, and also students, please remember that usually the question from this neuroregulation always comes in long question. Okay, so it will come for 10 marks or 15. Okay. So talking about the function of this aponestic center. So aponestic center is always involved in increasing the depth of inspiration by acting directly on dorsal respiratory group of neurons. And whereas the function of pontine center, pneumotaxic center, that pneumotaxic center is to control the medullary respiratory center, okay, particularly the DRG, okay. So it act through aponestic center and pneumotaxic center inhibit the aponestic center so that the dorsal group of neurons are inhibited, and because of this inspiration stop and expiration start. So in another word, we can say that the function of pneumotaxic center is switch off, okay. Because of this center only, there will be stoppage of inspiration and start of expiration will. Okay. So if the question over the this neural regulation will come, so you need to draw the flow chart, or you can also represent it by drawing the table, right? So medullary center we have here, then we have point line. Okay, then you can write that in pontine and in medullary, we have two parts. First, we have the DRG, then we have VRG. Then agonistic, we have pneumotaxic, we have. Okay. So first we're going to talk about the situation, where they are situated. So DRG situated in nucleus of Tractus solitarius, and the VRG situated in nucleus of ambiguous and retroambiguous. Okay, where aponestic center is situated in nuclei of reticular formation of lower pons, right? Then the situation of pneumotaxic center, that it is situated in nuclei of reticular formation of upper pons, right? Then type of neurons which are present. So these are inspiratory neurons, and here inspiratory neurons plus expiratory neurons both are present, and here inspiratory neurons are present. And which type of neurons are present in pneumotaxic? So here neurons of medial and suprabrachial nuclei. Okay. Now, what is the function of the center? Okay. So as already we have discussed that in medullary center, DRG function, that it generates inspiratory ramp. And also has autorhythmicity. And it remains always active. Okay. Then it is, the VRG is inactive during quiet breathing and active during forced breathing. Whereas aponestic center, the main function is prolonged inspiration and short expiration. And here in pneumotaxic center, as we have discussed, the function of the pneumotaxic is to switching between inspiration and expiration. Okay. So how it will switching? By inhibiting the inspiratory center through aponestic center. Okay. Now next we have the another important topic, that is hypoxia. Okay, and also students, please remember that hypoxia is a very important topic, and usually the question from hypoxia come in short also and in long question also. So what is hypoxia? Hypoxia means the reduced availability of oxygen to tissues. Okay, and another one more term is there, that is anoxia. Anoxia means, A means absent. So anoxia refers to absence of oxygen, and in older days the anoxia term was in use, since there is no possibility of total absence of oxygen in living condition. So use of this term is not being used right now. Okay, so what are the causes of hypoxia that we're going to discuss? Okay, so causes usually if oxygen tension in arterial blood, then oxygen carrying capacity of blood, then velocity of blood flow, and whenever there is utilization of oxygen by cells. Okay, and why this hypoxia occur, and what are the effect of hyperventilation? Okay, so whenever there is hyperventilation, so the hyperventilation can lead to the condition like, so hyperventilation can lead to the condition like hypoxia and hypercapnia, right? And if these conditions will be there, so they will lead the increase in rate and force of respiration, which can also lead dyspnea. Dyspnea means difficulty in breathing, and it will lead to the condition like lethargy, and the patient can also go in coma, which can lead to death. Okay, so this is all about the effect of hyperventilation that can occur. Okay, now we're going to discuss about the different types. So like as we can see in the slide, hypoxia, they are anemic, stagnant, and hypoxia. So now we're going to learn these hypoxia types in detail. So first we have hypoxic hypoxia. Okay, so hypoxia is occurs due to decreased oxygen content in blood, right? And talking about its causes, so various causes are there, like low oxygen tension, then due to respiratory disorders, respiratory disorder that are associated with decreased pulmonary ventilation, which does not allow intake of oxygen, enough oxygen. Then respiratory disorders which are associated with uh, inadequate oxygenation in lungs. Then also cardiac disorders are there, in which enough blood is not pumped. Okay. So these are the main causes, right, of hypoxia. Then next we have the another type, that is anemic hypoxia. So what is anemic hypoxia? Anemic hypoxia is a type of hypoxia that is caused by anemic condition. Okay. So here the oxygen availability is normal. Okay. So O2 is normal, but the blood is not able to take up the sufficient amount of oxygen due to anemia. So means here there will be decrease HB, hemoglobin, and decrease RBC. Okay. And what are the causes of anemic hypoxia? So various causes are there, like decreased number of RBC, then decreased hemoglobin content, then decrease or lead also due to information of altered hemoglobin, and whenever there is combination of HB with gases other than oxygen and carbon dioxide. Okay. So some poisonous gases are there. Okay. So due to that also an anemic hypoxia can occur. Then next we have the another type, that is stagnant hypoxia. Okay. So third we have stagnant hypoxia. Okay. So stagnant hypoxia, whenever there is decrease in velocity. Okay. So whenever there is decrease in velocity of blood. Okay. And here for the stagnant hypoxia, various causes are there, like vasospasm, then thrombosis, embolism, hemorrhage. Okay. And then another we have is histotoxic hypoxia. So as the name refers, histotoxic. Histo means something related to tissue and toxic means toxicity. Okay. So here histotoxic hypoxia, basically this is a type of hypoxia which is produced by inability of tissues to utilize O2. Okay. And what are the causes? The causes usually that is occurs in cyanide poisoning. So what is cyanide poisoning? These poison basically destroy the cellular oxidative enzyme. Okay. And there will be complete paralysis of cytochrome oxidase system. So even if oxygen is supplied properly, the tissues will not be able to utilize it. Okay. So this is all about the hypoxia type. So as we can see in this slide that hypoxia means there will be decrease in arterial PO2, the hemoglobin will be normal, and rate of blood flow to tissue is normal. Okay. Then anemic, the arterial PO2 is normal, hemoglobin will be low, and rate of blood flow is normal. Then in stagnant, arterial PO2 is normal, hemoglobin content will be normal, but rate of blood flow to tissue will be very less. Then in histotoxic, the arterial PO2, hemoglobin, and rate of blood flow to tissue will remain normal, and only they will be histotoxic hypoxia will occur due to poisoning of some kind of substance. Okay. Then also coming to treatment part, that if the hypoxia is there, then what can be the treatment that could be given to the patient. Okay. So here best treatment for hypoxia is oxygen therapy. Means treating the affected person with oxygen, and pure oxygen or oxygen combined with another gas is usually administered. Okay. So treatment is oxygen therapy. And how this oxygen therapy given? By placing patient head in a tent containing oxygen. Okay, and also if the hypoxia is there due to deficiency of like hemoglobin, then in that case blood transfusion can also. Okay, so this is all about the hypoxia, and I think we have completed all the topics. So we have completed with hypoxia, neuroregulation of breathing, surfactant, between restrictive and obstructive disease, dead space, lung volume capacity. So this is all for today's class, and I hope you find this class helpful. So thank you so much for watching my class.